Hi everyone, this video will walk you through the titration of vinegar experiment for Chemistry 10 at Santa Monica College. The video will only show you the experiment. The background and data analysis will be explained by your professor. You will need to record various data points in your lab report as the video goes through the experiment. We start by getting all the necessary chemical reagents and equipment for the experiment. The experiment requires a vinegar solution, a standardized sodium hydroxide solution, and a phenolphthalein indicator. Make sure you record the molarity of the sodium hydroxide in your lab report. We will use two measuring instruments, a 50 milliliter burette and a 5 milliliter volumetric pipette. The pipette is controlled using a pipette bulb that is shown here. We will do the experiment 3 times. The goal is to have multiple data sets so we can check our precision. We start by rinsing the burette over the sink using the ionized water, followed by the standardized sodium hydroxide solution. The goal is to make sure that the inside of the burette contains only sodium hydroxide. We then fill the burette up to the top with the standardized sodium hydroxide solution. Lastly, we flush the tip of the burette to remove the trapped air bubbles. The burette is now ready to use. Make sure you record the initial volume of sodium hydroxide for trial 1. The next step is to rinse the volumetric pipette. We do this by drawing in some deionized water using the bulb. After rinsing with water a couple of times, we repeat the rinsing using the vinegar solution. After the volumetric pipette is rinsed, we draw in exactly 5 milliliter of vinegar solution and transfer it into a 250 milliliter Erlenmeyer flask. After that, we add 20 milliliter of distilled water and 5 drops of the phenolphthalein indicator into the flask. We are now ready to titrate. We start titrating by adding the sodium hydroxide into the Erlenmeyer flask. You can see how the solution turns pink when the sodium hydroxide is added, but then disappears as it is mixed. Our goal is to titrate until the solution is light pink, which corresponds to the equivalence point. We can tell that we are approaching the equivalence point when the pink color takes longer to disappear when we swirl the flask. Here we are nearing the equivalence point, and with one more drop of sodium hydroxide, the solution turns pink. We now record the final volume of the sodium hydroxide for trial 1. This is the final color of the titrated solution about 1 minute after the end of the titration. We now repeat the titration 2 more times. Here's the initial volume of the sodium hydroxide for the second trial, and here's the final volume. And lastly, we have the initial volume for trial 3 and its final volume.